It's good to see everybody here, and thank you very much, Deborah, for inviting me. It's exciting to be able to collaborate with Kwantlen and this excellent institute that Deborah has set up, and the fantastic group of staff that she has. I've already spent the day with them yesterday, and they're really doing some cool things. So I will give you a quick overview of microbial biopesticides, just to sort of set the scene so that when Todd comes to talk to you and I go into more detail about the Lubulosa program, you've got an idea of what we're talking about. How does that go? At the bottom?
Bottom right.
That one. There we go. Okay. Okay, so I'm part of the Thomas Lab, which was set up at the Penn State University Department of Entomology about 5 years ago. Before that, I was working on the Lubulosa program for CABI Bioscience, which is an international organisation based in the UK, and we collaborate originally with Commonwealth countries, but it's become international, and we do a lot of developmental agriculture, or we did a lot of developmental agriculture research projects, and Lubulosa was part of that. Since Lubulosa, my husband, who is Matt Thomas, and I moved to Penn State University, and we're applying some of the techniques and technologies that were developed as part of Lubulosa to address other key pests outside of locusts and grasshoppers. So microorganisms used as biopesticides, where there are 5 groups: the bacteria, fungi, viruses, protozoa, a little less so, and nematodes. And there's been a long history of use of microbial organisms to control pest insects, and the first reference to that was by Pasteur and Leconte, who independently suggested the use of fungi as insecticides back in 1874. Mechnikov is another famous scientist back in the history of biocontrol. In 1879, he produced and released Metarhizium in a field experiment, and in fact Metarhizium anisoplii, as part of this taxonomic sort of system, is called Meched Sorokin, which is short for Mechnikov, because he was one of the first people to describe Metarhizium. Then biopesticides were taken over rather by Bt, and I'm sure you're all familiar with Bacillus thuringiensis. We had sporine, first of all, developed in the 1940s in France, going on to substantial development in the 1970s with the use of Bt. And in the 1980s, things really took off. But it wasn't until 1985 or 86 that pharmaceutical and agricultural chemical companies actually started to become interested in the fungi again, and in particular in Metarhizium and Bavaria. Bayer were very interested in a Metarhizium product which they found they could produce in liquid culture, and that product never actually was developed by Bayer, but the strain that they were developing was taken over by another company then called Earth Biosciences, and has gone through many iterations and is now what you might discover at some point. I think it's available on the market as Met52, produced by Novozymes. It's the same isolate that was originally developed by Bayer, but they weren't so successful. But hopefully Novozymes will have a little more luck with that particular product. And then from 1985 onwards, there has been an increasing interest, public interest and scientific interest, in development of biopesticides. Currently, biopesticides are used for insect, weed, nematode and disease control. Fungi are prevalent in all of these products, and they're used as replacements for harmful chemicals and essential components in IPM, and also good for resistance management. So looking at the fungi in more detail, there are a range of different genera of fungi that attack insects. Entomothera is the top one there. You may have seen flies stuck to your window pane. This is the entomotheralian fungus that it produces epizootics, but it's a very difficult fungus to produce outside of the insect, the living insect. So it's not a very good candidate for a biopesticide, which you want to produce in the lab and then distribute inundatively or in a similar way to a chemical. But you've probably seen Entomothera If you look around, it's there. Cordyceps, if you heard of the zombie ants, there's a big zombie ant story. This took off last year. So cordyceps is a fungus that attacks ants, and that somehow it manipulates them before death to climb up to up a tree trunk to a certain height, where it then clamps on its mandibles and dies. And then the cordyceps fungus produces this cordyceps which comes out of the head of the ant, and it sprays fungal spores down on the ants which are passing on their track below. And this is the way that the fungus cycles in the environment. It's incredibly interesting, but again, it's really not much use for biological control or biopesticide development, because again, it's very difficult to grow in the lab. So better are the Picillomyces species, Bavaria bassiana and Metarhizium anisopliae. There are a number of products based on all of these three fungi. Picillomyces used for nematode control as well as insect control. Bavaria, you can see a fly affected by Bavaria, that's from our lab there, and then locusts with the Metarhizium anisopliae. You can see the spores on the outside. Bavaria is white and Metarhizium is green. That's basic taxonomy. How do they work? Well, the fungal spore lands on the cuticle of the insect, and it acts as a contact insecticide. It doesn't have to be ingested. The fungal spore will, or conidium as we call it, will germinate and produce an aprosorium on the surface of the insect, and then penetrate down through the cuticle, working its way through this penetration peg, through the epidermis, and then once it gets into the hemocoel of the insect, it starts growing as blastospores, which are these like individual sort of budding yeast-like structures. And over time, they go on to kill the insect. So that's how it works. It takes at least 3 days, more commonly much longer, to kill an insect using a fungal biopesticide. But this is okay, and we'll go through some of the details of what affects the time to kill and so on. In addition to virulence, there are also biotic and abiotic factors. So here's a list of some of the fungal products. I don't know, has anybody used a fungal biopesticide product here commercially? Yeah? What was the name? Shout out some names.
Metarhizium.
Metarhizium, and what was the product called?
It was one of my own.
One of yours, okay. A commercial product, anyone? Yeah. Met 52, great, that's the Novozymes one. Botanigard, that's the Bavaria that's produced in the US. Another one? Yeah, okay. Yes, there aren't so many registered in Canada. There are rather more in the US, but registration is a bit of an issue, and I understand that's something that Deborah and her team are trying to help with. Okay, so there are markets for biopesticides. Multinationals tend not to be so interested in biopesticides because they tend to be specific and for niche markets. Although there's some news about that, which we'll get onto later in the day. But they can serve a demand for local products and be tailor-made to local production systems. If you're producing locally, that reduces any storage and distribution issues that there may be, but to the large extent, storage and distribution has been sorted for these fungal biopesticides. You can use simple production technology, and if you're producing locally, you can save on foreign exchange. So these things don't have to be huge global companies. There are many advantages of biopesticides, and I've probably skipped a few here. Reduction in health risks and costs due to chemical poisoning. Environmental damage can be reduced. You get sustainable pest control due to preservation of natural enemies, and I'll go into some details on that from data from the Lubulosa project. You can create local employment opportunities, and if you're producing locally, you'll reduce expenditure on foreign exchange. So the current status is that we have considerable know-how on how to develop commercial biopesticide products, and there are a number of virus, fungus, nematode products available, marketed to a small extent by multinationals and mostly by small to medium enterprises. So there are some opportunities for regional-based niche markets for biopesticides, such as in BC, Canada, there is a decline in pesticide use, and there's a lot of pesticide resistance in a lot of the common pests that we're studying. Bedbugs and sea arid flies and house flies are three that I've identified that are in critical need of alternative products, alternative to chemicals, because of the build-up of resistance. Also, the larger chemical companies are not re-registering their chemical products for small niche market products, for niche market crop systems. So if it doesn't seem to make financial sense to them, they're not adding their chemicals to, or the crop system to their labels anymore. So you're getting a reduction in the number of chemicals that are actually available for niche market crop systems. And I've already said that we can do local production, but quality control is critical, and local production doesn't mean sloppy production. So there are measures to be implemented. So, have I spelled opportunities incorrectly there? I apologise if I have. So at Kwantlen, there's a fantastic opportunity from the institute that Deborah's running, where they can provide training and information on aspects of biopesticide development, such as the workshop today. They can assist with production, development, and bridging gaps, provide the product in advance of a company that wants to set up, of actually getting a production plant going themselves. Identification of promising biopesticide products, assistance with business plans, registration, and just fill in any other specialized research gaps that a company interested in developing biopesticides might need. So this is a fantastic opportunity here to actually push the barriers a bit further. So that's basically the overview of biopesticides with a focus on fungi. If there are any questions, I'll take them. Yes?
Being a non-specialist, excuse me if it's a silly question, but the mode of action of these pesticides, do they rely on application, or once you got a host population with a disease, is the disease self-sustaining to control those bugs?
It can be, and it depends on the pest system. It's not something that we rely on as one of the major features of a biopesticide. The ones that we use as commercial biopesticide products tend not to cycle greatly in the environment, although there will be some impact of that. But basically, we're looking at direct targeting or direct hit with a spray, or pick up by secondary contact, so by walking on a sprayed surface. And that's the principle that we use for controlling mosquitoes. We will spray walls, and the mosquitoes are light, and they acquire a dose. So that relies on that product, once sprayed, being persistent in the environment. But the actual cycling, that's not something we rely on too much. But I've got some details on that as part of the Lubulosa. Yes?
So these biofungicides, they only act as a contact. There's no other way to attack the pest or the disease?
Yeah, it's purely by contact. People have tried to look at whether ingestion, assists in the infection process, but it really doesn't. It's a contact insecticide, but it doesn't appear to matter where it contacts. That it's going to penetrate through the cuticle and start working if the isolate is virulent to the pest that you're interested in. And there's a degree of isolate selection that's important when you're looking at this. Not all isolates kill all insects, so there's a selection process. Yeah?
Have you noticed that it depends on the life stage of the insects for effectiveness, or like that's also specific?
Yeah, that's very much part of the development process, and it does depend on the pest as to whether they're susceptible at different life stages or not. Bed bugs, any life stage, apart from the egg. We haven't tried eggs, but yeah, and any instar is susceptible with the bed bug. But yeah, and it also depends operationally with locusts. You want to target them early rather than late, just to prevent them going on to reproduce. So, yeah.
Good questions.
Yeah.
I wanted to just make one point, and Nina said it, but I don't know if it just went over your head, that, what did you say, the use or the sales of pesticides is decreasing?
Well, it's all over the place, I think.
Yeah.
But yeah.
But in fact, biopesticide sales are increasing. That's the growth area
Yes.
in pesticides right now.
Yes.
It's still small, but it's a growing area.
Yeah.
Thank you.
Okay.
Let's
So
do the
is
Lubulosa story.
Lubulosa, straight into Lubulosa. Okay. Let's do that. That's fine.
All right?
Yep.
Okay.
Let's